What a great crowd this evening for such a special occasion. I'm Rick Lifton, the president of Rockefeller University, and I'm truly delighted to welcome all of you to the 22nd annual presentation of the Pearl Meister Greengard Prize. It's a real privilege to have you all here to one of the university's most cherished events, where we honor women scientists whose discoveries have profoundly shaped modern biomedical research. Over these 22 years, 28 remarkable scientists have received the Pearl Meister Greengard Prize, and tonight we're delighted to recognize our 29th distinguished honoree, Dr. Maria Jasin. Maria is an esteemed colleague, and in the Small World department, she just so happens to conduct her groundbreaking research across York Avenue at Memorial Sloan Kettering Cancer Center, where she opened her lab in 1990. Before we begin tonight's program, I'd like to note that Rockefeller is approaching a very special milestone. In 2026, the university will celebrate the 125th anniversary of our founding and the 70th anniversary of the founding of the David Rockefeller Graduate Program. So for 125 years, Rockefeller has been a world leader in biomedical discovery, reshaping modern science and advancing human health. Founded in 1901 as the nation's first institution devoted exclusively to biomedical research, Rockefeller pioneered a model of discovery rooted in pursuing curiosity, collaboration, and excellence. Our achievements connect early triumphs against infectious disease to transformative insights into the molecular basis of life today. In recognition of these advances, 26 Rockefeller scientists, 10% of the professors in our history, have received Nobel Prizes, and 26 have received Lasker Awards, a testament to the extraordinary impact of our faculty's discoveries. Among our Nobel laureates is the late Paul Greengard, a pioneering neuroscientist and co-founder of the Pearl Meister Greengard Prize. Paul received the Nobel Prize in Physiology or Medicine in the year 2000, and for his discoveries about the mechanisms of signal transduction in the brain that regulate the activity of neurons. His work has elucidated causes of neurological and psychiatric disorders, including Parkinson's disease, depression, schizophrenia, and Alzheimer's disease. Paul and his wife, the renowned sculptor Ursula von Ruydingsvard, established the Pearl Meister Greengard Prize with his monetary share of the Nobel Award and named this prize after Paul's mother, who died giving birth to him. Paul and Ursula were guided by their belief that women scientists had not been appropriately recognized for their contributions to science, and they set out to change this. Their vision and generosity inspired Friends of the University to add their own support to this prize, which now includes an honorarium of $100,000. We are delighted that Ursula is here with us tonight, along with members of the Greengard family. Ursula is back in the back. Please wave and be acknowledged. Thank you, Ursula. Ursula, we're delighted that you're here, and thank you so much for continuing to carry forward this remarkable legacy of this award. So the prize recipients have been true trailblazers who have pioneered advances in molecular biology, chemistry, cancer research, immunology, biochemistry, genetics, biophysics, and neuroscience. To date, 4 recipients, Elizabeth Blackburn, Carol Greider, Jennifer Doudna, and Catalin Carrico, have gone on to win their own Nobel Prizes. You will have seen images of all of the recipients of this award etched in glass in the exhibit along the window in the Abbey Aldrich Reception Hall. Completed in 2021, this installation stands as a lasting tribute to these extraordinary scientists. I'd like to take the opportunity to acknowledge my colleagues on the Pearl Meister Greengard Prize Selection Committee, several of whom are here in the audience with us tonight. We're fortunate to have such a distinguished group of leaders taking part in the selection process. The complete committee list can be found in your program. So tonight, we're recognizing Dr. Maria Jasin, a renowned cell biologist and developmental biologist who did the first experiments showing that double-strand DNA breaks in mammalian chromosomes could be introduced at specific sites and exploited to edit the DNA sequence. a discovery that laid the groundwork for the development of CRISPR and other gene editing methods to come many years later. She also solved the mystery of how inherited mutations in the genes BRCA1 and 2 cause predisposition to breast and ovarian cancer. Maria showed that these genes are required for error-free repair of double-strand DNA breaks, and their mutation increases the DNA mutation rate, thereby increasing the likelihood of mutations that lead to cancer development. Her discovery led to the development of drugs that selectively increase the production of double-strand breaks in these cancer cells, resulting in their death. These drugs have benefited countless patients with mutations in the BRCA and related genes. Maria's work has been highly original and impactful, and we look forward to hearing more about her remarkable career tonight. I'm delighted to extend a special welcome to Maria's sister, Joanne, who's here with us tonight, as well as many other colleagues and friends. Thank you all so much for joining us. So, as part of the Pearl Meister Greengard Prize presentation, we have a special tradition of inviting a distinguished woman from a different field to present the prize. This year, we're thrilled to welcome Wendy Evans Joseph, a visionary architect whose work has profoundly shaped the way we experience cultural and educational spaces. From museums to academic institutions, her designs blend functionality, beauty, and purpose, creating environments that inspire learning, reflection, and connection. Wendy's a graduate of the University of Pennsylvania and the Harvard Graduate School of Design, where she was valedictorian and received both the Henry Adams Medal and the James Templeton Kelly Prize. Wendy began her career at Pei, Cobb, Freed & Partners. At the firm, she contributed to major civil and cultural projects, including the Boston Federal Courthouse, the U.S. Holocaust Memorial Museum in Washington, D.C., where she was lead designer. During this tenure, Wendy also received the prestigious Rome Prize in Architecture from the American Academy in Rome. In 1998, she established her own firm named Studio Joseph. One of Wendy's early commissions was right here at Rockefeller. When Torsten Wiesel was president, he asked Wendy to design the pedestrian bridge over 63rd Street that connects the campus's Weiss Research Building to scholars' residence, where many fellows and faculty live. This bridge permits Rockefeller's scientists and their children to blissfully walk to campus, bypassing the heavy traffic coming off the FDR Drive that jams the corner of York and 63rd Street, creating a notable pedestrian hazard. The bridge transcends its utilitarian function, achieving a distinct elegance through a V-shaped steel tower that supports a Y-shaped deck. The bridge was so popular and well received, it's colloquially regarded as the Torsten Wiesel Bridge. Torsten is here in the audience this evening, and we thank both you and Wendy for your wonderful collaboration. But in one of these unusual twists of fate, I note that our other honoree tonight is actually a resident of Scholars Hall and has been in Scholars since 1990, I presume. And so Maria has directly benefited from Wendy's creation. So recognizing both of them tonight is a wonderful coincidence. Wendy's studio has led other amazing projects, including the Women's Museum in Dallas, the renovation of Snug Harbor Cultural Center in Staten Island, and the London Mithraeum, a reconstructed Roman temple. She has also designed exhibits for the National Museum of the American Indian, the Library of Congress, and the Cooper Hewitt Smithsonian Design Museum. At her alma mater, UPenn, Wendy also established a scholarship to fund summer travel for architecture students to explore natural and constructed environments throughout the world. A fellow of the American Institute of Architects, Wendy has served as chair of its National Committee on Design. She has led the Architectural League of New York and currently serves as president of the National Academy of Design. In 2023, she was named a Fellow of the Society for Experiential Graphic Design in recognition of her contributions to immersive and equitable design. We're honored to have Wendy here with us tonight, not only as a distinguished architect, a champion of innovation, inclusion, and excellence, but also as a member of the Rockefeller University family, as the spouse of our own professor, Jeff Ravitch, also renowned for his innovations in immunology and cancer research. Please join me in welcoming Wendy Evans Joseph to the stage.
Wow, thank you. Thank you, President Lifton, and thank you for this very gracious welcome and kind introduction. I am so happy to be here for the Pearl Meister Greengard Prize Selection Committee's choice of Dr. Maria Jasin. And thank you very much for inviting me to participate in this presentation. It's a real honor to celebrate, Dr. Jasin, your work as a woman of science and your discoveries that frankly resonate beyond authorship. As you heard from Dr. Lifton, I'm an architect. For me, it's personal. It's not just what I do, but who I am. Architecture is my way of navigating the world and contributing to society. Architecture, like science, is a field that has traditionally been male-dominated. But to be a woman in these professions is not in and of itself a goal. And to a degree, neither is it a distinction. The question is how our identity, our passion for discovery, might inform and reinforce the work we do. I've been asked to reflect on my experience. Start at the beginning. I didn't know much about architecture growing up, but what was I doing? I was doing math problems for fun. I built furniture with my father in our basement woodshop. And yes, I bled radiators. When I told my father I decided to study architecture in college, he was thrilled. One problem. He thought architect was a boy word. Waiter, waitress. Actor, actress. Architectress. No. So he smiled and called me girl architect. And that term endearment shaped my professional path. I decided if I were going to be an architect, I would go big, go strong. No girly stuff for me. I began my career in the offices of I.M. Pei, designing skyscrapers, courthouses, and large-scale infrastructure projects. I wanted to be taken seriously, which meant being in the most male-dominated sector of the male-dominated profession. But it was never enough for me. I wanted to not just be there, but I wanted more. I wanted to lead, not just as a girl architect, but as an architect. After about a decade in these realms, everything changed when I was assigned to the United States Holocaust Memorial Museum in Washington. My role included talking to survivors, ghetto fighters, partisans. I listened to their stories. I traveled to the concentration camps. I started to design, not with program, but with narrative. I wanted to design to promote healing and to remember. Every architectural element, its materiality and form, contributed to metaphoric storytelling. When the museum opened, I recognized that I could not go back to the commercial world, to buildings that are mute. So I left the Pei office in search of meaning. Fortuitously, Governor Ann Richards was planning a women's history museum in Dallas. Her team read an article about me in, and how perfect is this, Working Woman magazine. So they hired me and later explained that there actually were no other women architects. And then in a tour de force of politics and architecture and education and all this Texas grit, we built the first ever women's museum. And we built it with an entirely female-led team: design, construction, historic preservation. And at the opening, Gloria Gaynor came and sang "I Will Survive." And I laughed to myself. I thought, okay, I can finally embrace my girl perspective. It's working while fulfilling my ideas about meaning and just making it for the community. Now, not long after, I found myself in a dinner conversation about a bridge with then President Torsten Wiesel. And armed with experience that was required to identify, let's say, an unexpected structural concept, I was able to cut costs in half. But what Dr. Wiesel and I fought for was the community. We created a structural link between home and work that was safe. But the bridge became more than that. It became the underpinning of the idea of Rockefeller's life science connection. So now, 2 decades later, my practice is exclusively one of community education and places that forge social bonding. I focus on public libraries, welcome centers, collaborations with museums to create content-driven learning experiences. But this year, work in the architectural realm has been ambushed by misinformation. The reemergence of the neoclassical might seem superficial or even comical, but it personifies a return not to any real past, but to an idealized past of emperors and peasants. Along with social anachronisms like ballrooms, we're seeing narrower definitions of cultural expression and outright censorship. As a society, we understand that there is inequity. There are racial, economic, and other forms of disparity due to historic privilege. How we seize this moment to adjust course is what defines us. Consistent ownership of the problem requires allocating resources to open pathways for education. We need to help those who lack opportunity. So what does that mean at this moment in time, both the omissions and the attempts to rectify the situation? Recent politics censor the very words we need to communicate about our work in science and the arts. Those words on the administration's list include gender, community equity, cultural heritage, and now it gets juicy. Female, females, feminism, and to the point of our celebration today, the word woman. We expect our leaders to have a basic level of understanding. Scientists make transgenic mice, not transgender mice. Antibody diversity is not a social challenge. New executive orders not only limit artistic and architectural creativity, but they also increase cost, preventing our abilities to address current and changing needs. And they inhibit our freedom to create places and help underserved communities. We expect our leaders to understand that as a nation, our history is beautiful, even with its complexity. Only by owning that history and not creating myths or rewriting it will we be able to move forward. The number of women in science since Dr. Jasin began her career is indeed higher, but not dramatically so. What has emerged is a different and equal troubling issue. Women in the field are not consistently recognized as frequently as their male peers. The late Paul Greengard and Ursula von Rydingsvard saw, rightly, that we have to make a change and not wait for it. I've had the privilege of knowing the benefactors of this prize, a remarkable couple whose work and contributions transcend professional boundaries. Paul Greengard was a humanist, and he was proud of the changes in science that his mother was not able to enjoy. Ursula expresses her hopes for the future, creating provocative and beautiful sculptures. They ask us as women to be proud of how far we have come, but they expect us to harness our power to ensure continued advancement. Let us magnify Dr. Jasin's impact by celebrating her most deserved Pearl Meister Greengard Prize. And on behalf of the Distinguished Selection Committee and all of us here today, we congratulate you on this award. Thank you.
you, Wendy. That was beautiful. Thank you, Wendy, for those beautiful comments. And I'm reminded that geneticists everywhere lie in fear of having to use the words cis and trans for mutations on different chromosomes or the same chromosomes in their grant applications. So it's now my great pleasure to introduce Dr. Maria Jasin, our Pearl Meister Greengard Prize honoree this evening. She holds the William E. Snee Chair at the Memorial Sloan Kettering Cancer Center. Maria was born in Detroit, and she spent much of her early childhood there. Her mother died of cancer when Maria was only 8 years old, and her father moved the family to Florida soon afterward. He had not gone far in his own formal education, but he recognized that his daughters were really smart, and he respected their intelligence. He bought a set of encyclopedias to feed Maria's curiosity. It was the mid-1960s, and the United States was swept up in the space race. Maria immersed herself in Time Life books about astronomy, physics, and biology. She excelled academically and graduated from high school as her class valedictorian. At Florida Atlantic University, she majored in biology and became captivated by the blossoming field of molecular biology. In college, she was again at the top of her class, and she was accepted into graduate school at MIT in 1978. When she arrived there, she immediately recognized that her academic background did not match that of her classmates, many of whom hailed from elite research universities and already had conducted considerable research. She realized she would have to really dig in to catch up, and boy did she ever. By the time she finished her PhD, she had published high-impact first author papers in Cell and Nature, leading journals in the world of biomedical science. As a graduate student in Paul Schimmel's lab at MIT, she harnessed developing recombinant DNA methodologies to do one of the first structure-function analyses of a protein, the E. coli alanine tRNA synthetase gene. By exploiting the bacterium's capacity to perform recombination, the swapping of pieces of DNA, she invented a way to replace a stretch of bacterial DNA with any desired sequence. Realizing the power of this approach, called targeted mutagenesis, to understand gene function, she wanted to find a way to similarly modify mammalian DNA. According to beliefs of the time, homologous recombination occurred at measurable levels only in cells that were on their way to become sperm or eggs in the germ cells. In other cells of the body, the process was absent or rarely occurred or just couldn't be measured. But Maria had a different idea. In yeast cells, double-strand breaks in DNA were known to stimulate homologous recombination, also called homology-directed repair, at the DNA cleavage site. Maybe she thought she could nudge mammalian cells to do the same thing. To that end, she went to the University of Zurich as a postdoc to work with Walter Schaffner on a mammalian virus, SV40. She showed that double-strand DNA breaks can indeed enhance the virus's ability to acquire homologous, that is, highly similar DNA sequences from the chromosome of a cell that the virus has infected. She then extended these findings by going to Paul Berg's lab at Stanford. And in 1990, she moved to the East Coast to join the faculty at Memorial Sloan Kettering Cancer Center, where her new lab geared up to make what would turn out to be a breakthrough discovery. Because sites of double-strand breaks stimulate homologous recombination in yeast chromosomes, she needed a way to create a double-strand break in a mammalian chromosome at a unique sequence. She took advantage of a unique enzyme that only cuts DNA at the site of a specific sequence that is 18 base pairs in length. Such a sequence is so extremely rare that it was not found anywhere in the mouse genome. She then inserted the target sequence into mouse cells grown in culture and designed a clever experimental system that would allow her to detect modification of the chromosomal sequence by introducing a segment of DNA with a similar sequence that included an antibiotic resistance marker, potentially enabling homology-directed repair of the double-strand break that she would be able to recognize. She found that homologous recombination did in fact occur and was very frequent, much more so than people would have imagined. allowing her to introduce a specific change into a targeted site in the mouse genome. Furthermore, she had overturned dogma by showing that homologous recombination could occur efficiently in mammalian cells. The double-strand break could also induce repair by non-homologous end joining, essentially bashing the two ends at the break together, which frequently creates deletions and provides the basis for current methods that intentionally generate mutations in chromosomes. Maria's discoveries had unlocked the possibilities of DNA editing, chromosomal DNA editing. Her work took place nearly 20 years before the realization of this possibility, before the introduction of CRISPR-Cas9 editing in mammalian cells, which introduces double-strand breaks at desired sites in chromosomes to introduce the desired mutations into the chromosomes in living cells. Maria's experiments had laid the groundwork for what was to come many years later. But her findings would also soon launch her laboratory in new directions. In 1990, previous Pearl Meister Greengard Award winner Mary Claire King had mapped the location of a mutated gene, which turned out to be called BRCA1, whose inheritance markedly increased the risk of breast and ovarian cancer. The nature of that gene remained unknown until 1994, and by 1995, scientists had cloned the tumor suppressor genes, not just BRCA1, but also the related gene, BRCA2. But at the time they were identified, the normal biochemical functions of these genes was entirely unknown, but it was widely assumed that they held cellular growth in check. Work on these genes came up with a number of inconclusive or conflicting interpretations of their native functions. However, finding that BRCA1 and 2 co-localize with the recombination promoting protein RAD51 at sites of DNA damage, and the finding that cells deficient for BRCA1 or 2 were sensitive to DNA damaging agents prompted Maria's lab to investigate further. And they quickly showed that BRCA1 and 2 are in fact required for homology-directed repair, and loss of either of these genes increased the number of double-strand breaks, with cells relying on the error-prone non-homologous end joining mechanism to repair these double-strand breaks. The resulting increased mutation rate increased the likelihood of cancer-causing mutations occurring, thereby accounting for the increased risk of breast and ovarian cancer. Maria's discovery led to critical insight that BRCA1 mutant carriers without double-strand break repair, mutant cancers without double-strand break repair via homology-directed repair might be particularly vulnerable to increased production of double-strand breaks. This led to the development of PARP inhibitors. PARP is an enzyme that is involved in base excision repair, a step in single-stranded DNA repair, and inhibition of this enzyme leads to increased double-strand breaks, which are poorly repaired, resulting in selective cell death by apoptosis, while cells that still maintain one working copy of the BRCA genes are not so vulnerable. PARP inhibitors, when they went into the clinic, proved to be extremely effective in preferentially killing cancer cells that have knocked out BRCA1 or 2 or other genes required for homology-directed repair. They have become a mainstay for treatment of BRCA1 and 2 mutant cancers. In 2011, Maria also uncovered a second way in which BRCA1 and 2 protect DNA by preventing damage at stalled DNA replication forks. The replication fork is a structure that arises when a cell divides, playing a critical role in DNA replication and ensuring that stalled replication forks do not result in double-strand breaks. Maria has been increasingly well recognized for her contributions. She's an elected member of the American Philosophical Society, the National Academy of Sciences, the American Academy of Arts and Sciences, and the National Academy of Medicine, as well as a number of other impressive prizes. She has received many prizes in addition to the Pearl Meister Greengard Prize, including the Shaw Prize in Life Science and Medicine, the National Cancer Institute's Outstanding Investigator Award, and the Basser Global Prize for BRCA research. I'm proud to disclose, not having told Maria this before, but I was on the Shaw Prize jury when she received it in 2019. So this is not my first experience in reviewing Maria's work in detail, but every time it's an inspiration to read, and it's an amazing body of work. And so we're truly delighted this evening to pay tribute to Maria with the Pearl Meister Greengard Prize. Please join me now and to the stage. So I have to read this now. So I have here the citation that we are presenting to her tonight, which I am now obliged to read. Maria Jasin, you are being honored for discovering that, contrary to dogma, homology-directed repair of double-strand DNA breaks is present in many dividing mammalian cell types and can be effectively manipulated to edit chromosomal DNA sequences. These findings laid the groundwork for subsequent advances in targeted gene editing using CRISPR and other technologies that are in wide use in the laboratory and increasingly in therapeutics. Your discovery also implicated homology-directed repair in the maintenance of genomic integrity and the prevention of malignancies. This fundamental research led to your solving the mystery of how loss of BRCA1 and 2 genes promotes the development of breast and ovarian cancer. You showed that these genes are essential for homology-directed repair, and in their absence, double-strand breaks are repaired by other means that are much more mutagenic. Your critical discovery led to the insight that these BRCA-deficient cancer cells are especially vulnerable when other DNA pathways or DNA repair pathways are blocked. This knowledge resulted in the development of PARP inhibitors, which increase the formation of double-strand breaks and trigger selective cell death. These drugs benefit cancer patients the world over who have mutations in these genes. With unusual insight, intuition, determination, and the rigorous design and execution of incisive experiments, you have contributed massively to the challenge of conventional thinking in science, making fundamental discoveries about nature and applying this knowledge to benefit humankind. You've set the highest standard for the community of scientists by demonstrating how ingenuity, audacity, and courage can fuel discoveries with far-reaching impact. Today, in presenting you with the 2025 Pearl Meister Greengard Prize, we salute your brilliant science and thank you for your many contributions to humankind. So on behalf of our presenter this evening, Wendy Evans Joseph, the distinguished jury of the Pearl Meister Greengard Prize, and all of us here this evening, we congratulate you on this award. Congratulations.
Wow, that was an amazing introduction. Thank you so much, Rick, and thank you to Wendy as well. I'm deeply touched to be receiving this honor coming from the faculty of my neighboring institution. I want to thank members of the selection committee, including the Rockefeller women on the committee, Titia DeLonga, Corey Bargman, and Leslie Vossel, who are themselves so deserving of this award, but traditionally excluded from receiving it. This award is very special in its provenance. As was mentioned, Paul Greengard used his Nobel Prize monies to fund the award to recognize outstanding women scientists, while also remembering his mother, who died giving him birth. He saw that his mother's death at so young an age almost removed her from memory, but now her legacy is very much alive in this award. This is very touching to me, given that, as Rick mentioned, my mother also died when I was young, and I often feel that she is also forgotten. I want to thank Paul and Ursula for setting up this award and the continued support of the Greengard family. I want, of course, to thank those that have nurtured my scientific career, especially Paul Schimmel, Walter Schaffner, Paul Berg, who often gave me the freedom to pursue my own interests. That was really important in my career development. And also, of course, my many friends and colleagues who have impacted my career. Of course, I'm indebted to my many lab members, many of whom are here, thanks to the proximity to my lab, who have done the work and brought it often into novel areas. I want to express a deep sense of gratitude to my sister Joanne, who has been wonderfully sisterly and supportive all of these years. Early on, she provided me with a model for how to pursue academics, something that was otherwise not present in our family. Finally, I have been living in Scholars on the Rockefeller campus since I arrived in New York, and during this time, the Rockefeller community has provided a welcoming and pleasant environment. I walk over Wendy's Bridge, beautiful bridge, very stunning. I guess Wendy's and Torsten's Bridge at 63rd Street every morning with this beautiful full river view, and then begin a lovely tree-lined walk for a couple of blocks before leaving the campus for Sloan Kettering. Moreover, I've been able to raise my 2 young puppies on the campus while they were getting their shots, and especially during these times, I could experience the friendliness of the Rockefeller family. So thank you all for the beautiful welcome this evening, and I am entirely in gratitude for this evening. Thank you.
Mr. Chair. Well, thank you so much, and again, congratulations. We have time for a discussion, and perhaps I'll start by asking you about your transition from growing up in Florida, going to college there, and then making the transition to MIT, a rather different environment.
Oh my gosh, I had never been to Boston or Cambridge before going. They didn't have interviews at that time, so it was completely cold coming there. I had to buy closed shoes. I had to buy a jacket, which was way too thin. So all these superficial things that you would think hit me pretty hard. But I knew right away it was just such a wonderful place to be at, that I had thoughts early on of being an engineer or maybe a physicist or this or that, mathematician. And so being at the center of technology, development, education was really thrilling. But it was hard. I remember one of our starting classes, I was completely clueless. I didn't know any of the language, and I had to drop that and move that to the second year. And you know, I knew that nobody else in the class was doing that. I could have felt maybe some shame at that or consternation, but I was glad the program allowed me to do that and allowed me time to catch up. And everybody in the graduate school at that time was very kind and very helpful, and giving me that extra time made it so that I could get to the qualifying exams and pass with flying colors. So I'm eternally grateful to the admissions committee and the graduate program at that time for really supporting my not only entrance but continued presence at MIT.
So how did you happen to pick bacterial genetics in Paul Schimmel's lab for your thesis?
Bacterial genetics was a big thing when I was a student. It was so interesting. We heard in our classes, we read a lot about Rec A and all these different phenomena. And there was one phenomena in particular that I was interested in, and Paul Schimmel was starting to work on that. But in the end, he had cloned one of the first bacterial genes himself, and he suggested a site-directed mutagenesis project on this. And you know, I'm almost ashamed to mention this, because for any student in the audience, you would think this is completely ancient history, but when I started graduate school, people were not bashing genes to look at domains and such. And that seemed like such an interesting problem and approach to understanding gene function. And so I have to say I was a little less interested perhaps in the tRNA synthetase angle on things, but just more interested in the overall approach to studying gene function. And so that was a really good lab for me to join, and Paul was a great mentor.
Among all of your accomplishments, you mentioned that one of your papers from Paul's lab was actually your favorite paper.
In my graduate career.
Well, then I would expect that then. It's not so surprising, perhaps.
Yeah, so you mentioned I had both a Nature paper, a first author Nature paper and Cell paper, and you know, I was a good student in the lab and worked well with Paul for doing these projects, but you know, in a way that was very much his work. He wrote the papers, and you know, we had daily discussions, but you know, doing site-directed mutagenesis on tRNA synthetase was his idea. But I did come to this stumbling block where I had the N-terminus of the gene being functional and the C-terminus of the gene being functional, and it made no sense. And the best way to explain it was that the temperature-sensitive allele of this essential gene was leaky, and so I really needed to make a clean knockout, and so that really, you know, obviously propelled me for my entire career and in the interest in modifying the genome, and so it was maybe not the first manipulation of the genome of E. coli through site-directed immunogenesis, but it was one of the first, and I think the first essential gene, and I had a conditional allele to make it work, and so I found it to be a very creative, interesting, and potentially powerful approach, and so that paper was in the Journal of Bacteriology, but I'm very pleased by it, I think, because I could, you know, it was more my work now and less Paul's.
Mm-hmm. That's great. So when did you catch the bug that, you know, you were going to go out and figure out how to modify a mammalian genome?
Well,
was that something that you developed by the time you started your postdoc?
Well, actually, my first postdoc in Zurich was with Walter Schaffner, as he mentioned, and he had just discovered transcription enhancers. And that was really amazing, coming from the bacterial field where the transcriptional elements were just upstream of the start site, to now we knew that the elements could be hundreds of kb away. So that was an amazing discovery. His lab had really high impact papers that drew me there. But I was not thinking of gene targeting, gene editing at that time because I didn't know how to approach it. But he was going away, gave me his grant and said, read this and figure out what you want to do. And within that grant was this tiny section on how putting in SV40 genomes could interact with a copy of the genome in the chromosome to make wild-type virus. And I thought, that's it. If mammalian cells can do that kind of homologous recombination, they can do other kinds of homologous recombination. So that was my entree into doing gene targeting, as we called it at that point.
Yeah, amazing. And from there, you went to the Berg lab and then on to Sloan Kettering, where you did these critical experiments.
Right. So the step was in the Schaffner lab, I was doing viral DNA, picking up sequences from the chromosome. In the Berg lab, I was putting sequences into the chromosome. But it was working. My approach was basically a promoter trap that worked very well, but very inefficiently still. On a per cell basis, it was inefficient. And so the idea, in both of those experiments, it was important to put a double-strand break in the plasmid DNA that I was putting in. So this came from the Shostak et al. Model of double-strand break repair, that the sequence that you want to change is the one that should undergo the damage. And so what I was doing as a postdoc, but that was in the plasmid DNA. For the chromosome, needed to have a way to make damage in the chromosome specifically. And this Bernard Dijon's lab at the Pasteur had purified this homing endonuclease, ISC1, 18 base pair recognition site, and that was perfect. Oh, and one thing, it was from yeast mitochondria, so they had to completely change the coding sequence to make it express in E. coli, but that meant it could also express in mammalian cells. So that was a perfect entree for me.
In the context of the time, in contrast, Mario Capecchi was doing targeted knockout of genes without the benefit of having specific targeting, way of targeting the target sequence.
Right. So my postdoc paper from the Berg lab came out exactly the same month as Mario Capecchi's positive negative selection paper, which was very impactful for embryonic stem cell editing. At that time, everyone was enthralled that you could do gene targeting, and even more so by the ability to take those embryonic stem cells and put them back into mice and make mice with that targeted mutation. So that was huge, the late 80s, early 1990s. And I think a lot of people thought that was about as good as you can get, but still those events on a per cell basis were not frequent. Yeah. You couldn't even immediately translate that to C. elegans or Drosophila. Mice were very special in having the cell type that had been developed that could go into animals, and that was just not possible. So we needed other methods to make things more efficient.
So projecting 30 years, 20, 30 years forward, if you go back to where you were when you did this first experiment that showed that you could efficiently target making these double-strand breaks, did you imagine where we would be today?
Oh, yes and no. So the paper came out in 94. In 95, I wrote a TIGS review, Trends in Genetics review, and I said, what we need is, so I was using the IEC1 endonuclease, a very defined site. What we need is a general way to make double-strand breaks in the genome to promote this anywhere you want. My thought at the time was certainly Watson-Crick base pairing provided the best entree to that. And I thought maybe people like Peter Dervin were making oligos with cleaving reagents, and that would be the route that we would go on. Never did I anticipate a natural system with CRISPR where the programmable nucleases were actually fairly rampant, different CRISPR systems. So I was envisaging something with Watson-Crick base pairing, but not RNA editing, RNA directed through a natural system. So what the yes part of that was, occasionally you have these insights, oh, this worked, like the experiments in Paul Berg's lab. Things are going to be different. People are going to be doing gene targeting often and frequently and be able to study genes. And then with the break experiments, people are going to be able to very efficiently modify the genome. The non-homologous end joining part of it was annoying, actually, but in fact, it turned out to be pretty useful and what a lot of people use today anyway. And, you know, we would always talk about being able to take something like that and bring it to therapeutics. Okay, our ultimate goal would be to do therapeutics, correct human disease. And I was not at all clear that that was going to happen in my lifetime. So I thought that was something way off in the future, but, you know, here we have it.
One of the things that stands out to me in reading your work is, in retrospect, the ideas seem like, why weren't you one of 100 people trying to do the same thing at the same time, both with making the break and introducing foreign DNA to a targeted site, and with the follow-up of what are BRCA1 and 2 doing, where it seems like nature was just playing to your strength all along.
Yeah, there's a real benefit and negativity too, but a real benefit to being kind of out there on your own, where there aren't a lot of people thinking about this. You know, about the break thing, the yeast people never needed to put breaks in the genome to do gene modification. It was so efficient that they didn't need to do that. So there was not that paradigm even coming from, you know, this important model organism. And, you know, as I mentioned, the people could start doing gene targeting inefficiently, but could do it and then make mice. And so I think when we first sent our paper out for publication, people didn't quite appreciate it or get it, at least the potential of it, because things were already working to some extent, without necessarily thinking that things could be much, much better. And so obviously people, there were other people interested, and then the zinc finger nuclease thing came about, which basically took a whole company to work out to get it to work efficiently. So there was this gradual buildup, and so when CRISPR came along, it was absolutely clear what to do, and luckily it worked beautifully.
Amazing story. We have time for questions from the audience. Yes, right here. And a microphone will come down momentarily. And the rest of you think about what questions you're dying to ask.
With the double-strand DNA editing that you've done, did anything relating to glycolysis have any kind of bearing with the work of editing?
Glycolysis, you said?
Yes.
We haven't looked into metabolism and its impact on double-strand break repair. You know, as we're learning, metabolism has an impact on many, many things, but that's nothing that we've ever really explored.
Congratulations with the gene editing.
Thank you.
It's right here.
I'm very struck in your discussion that you talked about efficiency and simplicity several times. And it strikes me that that's what I understand most mathematicians do in terms of figuring out not only the simplicity and the efficiency of it, but also the elegance of it. So I suppose I salute you for that, and I'm just wondering if you might comment about the similarities that you find from your own research versus what mathematicians continue to do.
Well, you know, obviously in school that would come up, and we always like the simple solution, especially if it gave rise to efficiency. But I can't claim any particular mathematical insights into applying this to my own work, except, you know, everybody always wants things to work efficiently, right? But I did really, it's true, it is a really simple concept in a way. You know, it was like the moment there was a way to introduce breaks anywhere in the genome or DNA damage anywhere in the genome, everybody did it. There was no, you know, with CRISPR, there was no delay you know, papers were coming out day by day, and now I don't know what the number is, but hundreds per day or thousands per day. So it was something that, you know, I know I would never have any holdover. And I'm sure this is exactly what Jennifer Dowden and others who worked in CRISPR wanted, was that it would be accessible to, you know, almost anybody. Sure enough, that's the case. So yeah, simplicity in the system was really major.
There's another question right here.
Thank you. It's extraordinary to hear how things coalesce in ways that you don't anticipate, and that seems to still be the importance of basic research.
Right.
And so with all of these new tools that have come into creation, apart from CRISPR, but we're talking about more of the computer world, how do you go about knowing the methodology that you've already created through using CRISPR as the organizing principle? What excites you now, and where do you see that ability to still think outside of the box that you alluded to earlier?
Well, I'll come back to bacterial genetics is incredible, and there are people here working on CRISPR systems from bacteria. You know, we're so, many of us using CRISPR, we use Cas9 and that's it, and it does its job fine. But that is, I don't even know if a needle in a haystack or what the exact term would be, but there are thousands or tens of thousands of different systems, and people are mining these to get all different variation or not even variations, brand new systems that you can do other things with. It's incredible. I wouldn't necessarily say I'd give up mammalian cells, but I would really recommend students thinking about their PhD or even going for a postdoc to consider bacterial systems. The immune system repertoire in bacteria is immense, and it's really, really interesting, as Sam Sternberg and Nama here know. So,
Question here.
It's intriguing that you had an 18 base target for your restriction enzyme, and I was wondering how such a thing would evolve. Did it come from a variant of CRISPR?
Not as far as I know. It's an interesting system. It's not actually a restriction enzyme, but something called a homing endonuclease. And it's a very selfish element. It's in mitochondria of yeast, and it's encoded within an intron. And if there isn't an intron present in another mitochondrial genome, that enzyme will go and cleave that mitochondrial genome at that 18 base pair site and stick itself in there. It's an incredibly selfish element with no known. Actually, I haven't looked into that for a while. There are related enzymes that are found in bacteria, and I think it probably is an early arisal in evolution. Those enzymes are not found, as far as I know, they haven't been found in mammalian cells at all or higher eukaryotes, but certainly in yeast.
I think we're at time, so.
Maybe one last question.
There's one more. I hope it's a good one.
No pressure.
Congratulations, Maria. So I remember reading the 1994 papers and trying to recreate the kind of time between that and the first studies to develop zinc finger nucleases from Chandrasegaran and from Dana Carroll. But you can't read those papers and relive what that was actually like to kind of see this field go from an idea and a first piece of data to then the first ZFN paper and then the next one. So I'm just wondering if you can try to bring us back to that moment. I mean, was it a big lightning bolt of insight that this was all going to change? Did it come out bit by bit?
Well,
what was that experience for you like? I'm really curious.
Yeah, I knew everything was going to change. I just didn't know how fast. And I think one thing that was so disappointing that we didn't get our paper in a high-profile journal is I thought we need minds working on this, and people should know that this is a problem to work on. Of course, there were some people working on zinc finger nucleases, trying to fuse zinc finger transcription domains to a nuclease domain. And I knew of that ongoing work, but I just did not see that working very well. I thought if I started doing that, my lab would end up being a technology lab, and I didn't want to do that. I was, around that time, BRCA1 and 2 were cloned, and there were these hints to look into that. And doing that biological medical problem was much more interesting to me than developing a technology which, you know, of course, would generalize my work. But, and in fact, as I mentioned, you know, it really took years to for this company to make it successful that other people could use it. But I certainly appreciate the pioneers in the field. But I think what took me much more, brought me much more excitement, although the 2005 zinc finger paper was very exciting, was when talons came along, because zinc fingers were problematic because they're complicated protein interactions, whereas the structure of TAL effectors showed that there could be these little alpha helical double loops going in and touching a certain base, and there was no interaction. So you could string along these. And if we never had CRISPR, we'd all be doing talons all the time, knocking out our gene of interest or even doing therapeutics with it pretty readily. What we couldn't do is screens and other things that you can only do with oligo approaches. So the talent story was just amazing to me. But of course, the CRISPR story is another level. Yeah.
That was a good question. Thank you. So perhaps I'll just close by commenting for a moment on your work is such a really tangible demonstration of curiosity-driven science without a specific goal in mind. I think your work had much more immediate impact on clinical medicine than many people might have anticipated. And as we talked about the development of PARP inhibitors from the discovery of the mechanisms of BRCA1 and 2 mutations, had profound impact on health. And in these challenging days of where's the next grant going to come from, maybe particularly from NCI these days, it's really gratifying to be able to spend a few minutes thinking about the power of science and the power of curiosity-driven science to have great impact on humankind. So for that, I'm grateful. I think all of us are grateful for your work, and we want to congratulate you again and thank you for the wonderful work that you've done. Congratulations. And thank you all for coming.